Computational gene prediction. In this lecture we're going to go through the process of going from a sequence of DNA to identifying features in that sequence, such as introns and exons, basically looking for where genes are within a DNA sequence. So if we think about a eukaryotic genome, the structure is relatively complex. We have promoters, we have exons, and we have introns. And of course the first exon will contain some sort of initiation codon, usually the AUG codon coding for a methionine, and then at the end of the final exon there will be a termination codon, a stop codon. Now, there may also be transcription factor binding sites upstream of the open reading frame, and we will talk about those in a separate lecture. So the problem is, how do we find where these introns and exons are? Here I've taken a segment of the human genome and annotated it with exons in the dark red orange, and introns in the yellow here, the pale yellow. But we also have a number of things that confuse this, because we have things like pseudogenes here, which are sections of DNA that look like they're coding for protein but actually aren't. And we have things like microsatellite sequences, we have transposons, and other types of repeats like line elements and sign elements, and these make the process of identifying the coding regions, which is what we're kind of really interested in, much more difficult. So taking a look at a single gene, this is Tri4, which is the gene encoding trypsinogen, the inactive precursor of trypsin. As with most eukaryotic genes, it's discontinuous. It consists of 5 exons in the red, split by 4 non-coding introns in the yellow, and these have some DNA transposons within them. To add to the complexity further, in some cases, and this is largely just limited to the antibodies and the T-cell receptors, we have discontinuous gene segments. So V28 and V29-1 are segments encoding the variable domains, or part of the variable domains, of the beta T-cell receptor protein. So these are not complete genes. They must be linked to other gene segments by DNA splicing. So remember, this is DNA splicing, splicing that occurs at the DNA level, not messenger RNA splicing. So this results in a permanent genome change during cell differentiation. So, as I said, what we're trying to do is go from a sequence of A's, T's, C's and G's to something much more informative, like this sort of diagram at the bottom. So we want to know which regions code for protein, where gene starts and ends, where the exon-intron boundaries are, and where the regulatory sequences are. So if we compare this problem of eukaryotic gene finding with prokaryotic, there are a number of differences. First of all, in prokaryotes, we're talking about relatively small genomes, somewhere between 0.5 and 5 megabase pairs. They have a high coding density, generally more than 90%, meaning that most of the DNA does code for proteins. And most significantly, we have no introns. This means that gene identification is generally relatively easy and has around about a 99% success rate. There are some problems. Because of the compact nature of these genomes, there are sometimes overlapping open reading frames, We can have short genes, and that can cause a problem because generally we put some sort of length threshold on looking for genes so that we don't just find random sequences between a start codon and a stop codon. And we also still need to find promoters, which is a more difficult part of the problem. In eukaryotes, on the other hand, we have much larger genomes, somewhere between 10 and 120,000 megabase pairs. We have a low coding density, less than 50%, and in the case of the human genome, around about 2 to 3% actually coding for proteins. And we have this much more complex intron-exon structure. This means that gene identification is complex and the overall accuracy is around 50%. Problems? Well, there are lots, and we're going to start discussing some of these. So, in terms of the methodology that we use, there are two major approaches: the ab initio methods and the similarity-based methods. So, ab initio methods use information about the DNA sequence, intrinsic information, if you like, that has been learnt from looking at other DNA sequences. The similarity-based methods look for similarity between a new piece of DNA and genomes that have already been annotated with where the introns and exons are. And there are then also integrated approaches which use bits of both type of method. Often an initial ab initio screen followed by use of similarity-based methods to refine the boundaries. As I said, ab initio methods make use of features of DNA rather than looking at similarities between DNA. So these sorts of features include things like GT-AG splice signals, and we'll come back to these in more detail later on, and things like sequence composition. There are certain features that we can look at. For example, the initial 5' exon will have a transcription start point with an upstream promoter, and it will end immediately before a GT splice signal. Internal exons will begin after AG and end before a GT splice signal, whereas the final exon will begin after an AG splice signal and will end with a stop codon and a signal for adding a poly-A tail. The similarity methods, or empirical methods, detect similarity with known coding regions. So we're looking for regions that are similar to known proteins. And we can also use regions that map to so-called expressed sequence tags. These are relatively short regions of sequence, generally up to maybe 600 base pairs long, from the ends, the 5' and 3' ends of messenger RNA. What that tells you then is that this is a real region of DNA that is encoding a protein, because if we have our RNA, we know that that in turn is going to code for protein. So if we look at the beginnings and ends of a section of RNA, then we know that that bit, when mapped onto the DNA, is going to be a real coding region. Now, in fact, ESTs are used less these days because of the availability of high throughput, much cheaper sequencing methods, so that we can actually do the sequencing of whole pieces of RNA instead of just the tags. So at that point we're going to take a short break, and I would ask you to take a look at these two Wikipedia pages on gene prediction and a list of gene prediction software, just to familiarise yourself with the sorts of ideas that we're going to talk about.